Hello, my name is Shelby Harris, and today I'll be talking to you about the pathways of antigen presentation. So, antigen presentation is the process by which antigens are displayed on the surface of the cells to kickstart an immune response. So, antigens are presented on the major histocompatibility complex, or MHCs, and MHCs they actually come in three classes, but I'm not going to talk about class 3 in this video. But I am going to talk about MHC class 1s, which are found on all cells in the body. All nucleated cells in the body have MHC class 1 receptors on their surface. Another class is the MHC class 2, which is only found on professional antigen presenting cells like B cells, T cells, and dendritic cells. So only those that are the main players of the immune system are found to have MHC class 2s. MHC class 1s present antigens via the endogenous pathway, while MHC class 2 present antigens via the exogenous pathway. So to start off, I'm going to tell you a little bit about the MHCs, just a little overview of them. So over here we have MHC class 1 molecule, and as you notice, it's broken into two separate proteins. You have your alpha protein, which has three domains of alpha 1, alpha 2, alpha 3, and then down there in the bottom right, you have your beta 2 microglobulin. It's two different proteins in here. As you notice, it only has one transmembrane segment and one cytoplasmic tail to anchor it into the membrane. And as you also notice, is the peptide binding cleft up here, it is closed. It's not over here where it's opened, and it's closed, and it's like smaller, I guess you could say, than that one. This one has some overlap in it. I'll talk about that in a second. But as you notice, for the most part, it has two separate proteins, and that's the class 1. So over here we have class 2, and as you notice, it's about two identical proteins. In here you have both the alpha chain and a beta chain that have two domains that are about the same size. It has two transmembrane segments and two cytoplasmic tails. Like I said before, the opening is opened, and it has some overlay. As Dr. Blair said, it's kind of like a hot dog in a mini bun. It will go over some. It can hold bigger antigens, essentially. And that's the MHC class 2 molecule, of course. This is on all cells. This is on professional antigen presenting cells, where you find these. So to start off, I'm going to talk about the endogenous pathway. And the endogenous pathway, like I said before, is antigen presentation on MHC class 1. And it's called the endogenous because it's endo. The antigen is already inside the cell. It is let's use a cell that's infected by a virus. That cell that's infected by the virus is already going to have viral proteins being synthesized in it because it's infected. This pathway involves a number of proteins, so ubiquitins, proteasomes, and the TAP proteins. Ubiquitins mark the viral proteins for destruction by the proteasomes, which of course digest the proteins, and the TAP proteins import the viral proteins that have been digested into the endoplasmic reticulum, the rough endoplasmic reticulum. And inside the rough endoplasmic reticulum is where the MHC class 1 receptor itself is built. It's where the proteins are synthesized, and that's where the digested viral protein will attach to the endoplasmic reticulum, and then it will travel to the Golgi apparatus, and then from the Golgi apparatus it will travel to the outside of the cell to be presented to T cells to start up an immune response. So here we have the pathway over here, the endogenous pathway. So here we have our viral protein that's inside the cell. It's endo. So it's going to be ubiquitinated. It's going to have ubiquitin tags on it. That's going to take it to the proteasome. The proteasome is going to digest the protein using enzymes and stuff like that. And once we have our digested protein here, given by our little dots, they're going to be imported into the rough ER over here by the TAP protein. And while it's in here, the MHC class 1 is going to be synthesized and built, and the digested proteins are going to attach to it, and it's going to move to the Golgi apparatus, and from the Golgi apparatus, it's going to move out to the outside of the cell to present to T cells to say, hey, I'm infected with a virus. I need to die. And the cytotoxic T cell is going to be like, okay, kill yourself. And then the next pathway we have to talk about is the exogenous pathway. So it's different from the first one because exogenous means that it's outside. extracellular viral proteins. So say we have a virus, but it hasn't infected any cells yet, but it's been picked up by, say, a dendritic cell. A dendritic cell is now possessing that on its MHC class 2s on the outside to either be read by a T cell or a B cell. So it's on MHC class 2, it's picked up by the cell, it's exo to it, extracellular. Like before, the antigen is going to be digested, but instead of being digested by a proteasome, it's going to be digested by an endosome using acid instead of enzymes. But it's still the MHC class 2 receptor is made in the rough ER, the same as before. But this time, when it's in the ER, it's going to be blocked by a protein called CLIP, and that's going to block the receptor of the MHC class 2. So intracellular antigens can't be presented by the MHC class 2 by that way. So it's blocking it, and it's going to come out, and it's going to travel to the Golgi apparatus while the antigen is being digested by an endosome elsewhere in the cell. It's going to travel to the Golgi apparatus. Inside the Golgi apparatus, a protein called HLA-DM is going to remove the clip, and then it's going to fuse with the endosome, and the endosome is going to allow the binding of the antigen to the MHC class 2 receptor, which will then move to the outside of the cell to be presented. So here we have the pathway. So starting up here in the rough ER, we have our class 2 MHC being built, synthesized. It's going to move down here, and it's not shown in here, but we have clip, and it's going to the clip is just going to block the receptor right here, so these can't come over here and bind. So it's going to move into here to Golgi apparatus. Clip is going to be removed by the HLA-DM, and then the antigen, the blob that's going to contain the antigen, is going to fuse with the blob that's going to contain the MHC class 2 receptor, and they're going to bind, and then it's going to move to the outside where it's being presented. And of course, here is our antigen coming into the endosome that is going to be digested by acid into digested antigens to be presented. So that was our two pathways, and that's how antigens are presented on the cell. But one last thing I want to talk to you about is cross-presentation. And cross-presentation is essentially where antigens can cross the pathways. So in cross-presentation, exogenous antigens can be displayed on MHC class 1s instead of MHC class 2s. And this allows non-professional presenting cells to display extracellular antigens so that cytotoxic T cells may react with them. So a cell might not have regular MHC class 2 receptors like it can make, but it can pick up extracellular antigens without being infected and present them on their MHC class 1, so a T cell can say that and say, there's something here. So it cuts out the fact that if we only relied on our regular pathways, we would have to wait for a professional antigen presenting cell to get infected before we can really kickstart the real immune response with the cytotoxic helper cells and stuff like that, like our real immune response. So it kind of makes it speeds it up a little bit by allowing a non-professional presenting cell to present those. One thing I found while reading up about this is cancer immunotherapy vaccines and dendritic cells. So I read that they have made cancer tumor vaccines by using the genes from the anti-tumors to target specific antigen dendritic cells, which are professional antigen presenting cells, to go to them and where they'll go into the lymph nodes, and where instead of presenting stuff on their MHC class 1s, they'll present stuff on their MHC class 2s, and there they'll go activating the cascade of the T helper cells and the cytotoxic T cells and stuff like that, instead of just waiting for one T cell to find it. So that increases anti-tumor immunity. So because cancer cells, they can hide out in the body really well, they find a long time they have a way of avoiding the immune system and stuff like that. So that's going to speed up the immune system and be able to find those tumors and have your immune system attack it as point of cancer treatment. So I thought that was kind of cool. So here's a picture of cross presentation. So here we have your regular endogenous pathway where we have endogenous antigens being digested and added to MHC class 1s being brought out. But here we have here we have our exogenous where we have our MHC with clip being brought into here with the exogenous antigen being brought out. But here we have where our endosome containing our exogenous antigen can come up here and fuse with the rough endoplasmic reticulum and release antigens into here, like how the tap is importing antigens into here. Well, now here we have our MHC class 2 is still blocked, so it's going to be forced to bind to the MHC class 1 to where it will come out to the outside and present to T cells. So that's how it will bypass only being presented on class 2 molecules, because it will come up here to where this is free, but this is not, and it will bind. And so I thought that was kind of cool. So here's the end of my presentation. I hope you liked it. Thank you.